Are you taking AP Biology? Well, you're in the right place because today we're going to do my FRQ Friday. Yeah, I know it's not Friday. But so traditionally, every Friday leading up to the AP exam, I go live and I do FRQ sessions. So I wanted to come live today so that I could go through the 2022 AP Bio exam questions with you. So without further ado, wait, my camera's not on. Oh, there it is. Okay. So real fast, who am I and why am I coming live to you on this beautiful little YouTube account? So my name is Tiffany Jones. And I have been teaching AP Biology for, this is my 12th year. I am an AP reader, which means I do fly out to Kansas City every single summer and I grade your exams. So I kind of know how lovely your handwriting is. And then a bachelor's in biology and a specialist in instructional technology, which is why if you were already following me, you already know like AP Bio Penguins, I'm all about like using technology to teach you between the quizzes, between using, Instagram, TikTok, all these different platforms to try to help you out. So why do I call students penguins? Well, because of course, penguins have their nice little cute little tuxedos. And so as penguins, you're now dressed for success and you are going to rock this exam that's in 11 days. Oh my God. So May 10th, you all know this, at noon is when we are going to be taking the AP exam and you are going to rock this because you are a penguin. So real quick, before we kind of get into these FRQs, I wanted to give you a little bit of breakdown, a little background on this. So on the exam, you're going to have a total of 6 different FRQs, okay? So the first 2 FRQs will be your long FRQs. So in the CED, it tells you specifically, I'm sorry, I'm getting over a little cold. It tells you specifically what the topics are, like how they're going to ask you these questions, and they tell you the points. So top questions 1 and 2 are going to be 8 to 10 points. Together, they're going to total 18 points. And then your 4 short FRQs are each going to be worth 4 points. And what's going to be noticing is A, B, C, D. So each letter is going to be worth 1 point. And so it's a total of 16 points for your short FRQs. So overall, some tips when we were dealing with FRQs. You want to read the question. I don't know if you heard me. I said read the question. Oh, wait, did you not hear me? Read the question, okay? Make sure you're answering the question that they're asking you. I know that sometimes as you're writing, you're like, oh my God, yes, I'm writing all this stuff. I know all this information. Look at all this biology I know. And you've completely not answered the question. You've just gone on your own little tangent because you're amazing and you know so much biology, but you didn't answer the question that they were asked. So periodically look back at the question, make sure that you're answering the question that's being asked of you. And somebody just asked how long this is going to be. It's 2 different sessions that I combined into one. I'm really just going to run through the session. I'm going to run through all the FRQs. I'm going to make sure that I answer as much of it as I can. I'm going to try not to speed run through it. I want to make sure that you're understanding the different questions and where we can get our information from, and you'll learn as you go through this. So my goal is to stay within the hour, but I like to talk. So label your sections. So when you're answering part A, just write A and then write it out. B, write it out. C, so on and so forth. And answer in the way that you know it. If you know part C, answer that first. Write out C and answer the question. If you know D, write out D. Then if you know A, it doesn't matter to us, the readers, in which way you answer it. We just want to know, what are you answering? So we're a little bit kind of helps us, okay? Don't restate the question, okay? We know what the question is. We know what the prompt says. Restating the stuff that's given to you is not going to get you points, okay? And then if they ask you to give 2 examples of something, like name 2 of these. Naming 3 doesn't get you extra points. It doesn't help if you had one of the other ones wrong. We only grade the first 2 that we will see. Okay. Beware contradictions. Don't tell me that the water is going to move into the cell and then tell me because the cell shriveled. Like that's contradicting. That's going against the statement you just made. So be very careful that you don't go against the other. Okay. Use your diagrams and of course use your diagrams. Okay. And no, you don't have to answer the questions in order. If you know question 5, answer 5, then go 2, then go 5, 6. It doesn't matter to us in which order. Answer in the order that you know the information. Define your terms. Okay. Sometimes just writing that little bit of extra thing to show us that you know what you're talking about helps to get the points. Okay. So in terms of like your graphing, like number 2 is always going to be a graphing question. So label your axes, okay? Make sure you have the correct scaling. Traditionally, that is one point. If you can literally just label the axes, which is just looking at the chart and writing the exact thing that's in the chart. Sorry, y'all. And then like having the correct scale and having the correct number of like letters. I'm sorry. Like if you make a block 5, then don't make 2 blocks 5. Like you have to make sure that every block is only 5. Okay. So make sure you have your scaling. Write as clearly and legibly as possible. Okay. I know you have 90 minutes to write 60 to write 6 FRQs, and yes, there's going to be a cram session. It's next weekend. So make sure you write clearly and legibly. I know you're Russian, but it's very hard for us to grade it if we can't read it. Okay. When you say something wrong, just go ahead and just put a single line through it to cross it out. You don't have to scribble it. You don't have to do anything crazy. Just one line straight through it, and we understand that you didn't mean to say it. Okay. You're supposed to write in pen, but you and I both know that sometimes you got to do that graph in pencil, or you didn't realize you were using that pencil, and oh my God, I'm on the second question and I never went back to my pen. Y'all, it's okay. Just switch it over, use the pen, and get going. Okay. And then just make sure that your writing pencil doesn't bleed. We're grading scanned in responses. So they'll literally take your FRQ book and they'll scan it in the machine. And we're looking at a computer image of your FRQ. So being able to not have it bleed through and being only able to see the one side helps us a lot. Okay. So in terms of timing, you have about 25 minutes for your longs and 10 minutes for your shorts. Okay. Your goal is to write the long ones in 20 minutes and your short ones in 8. Why would you do this? It gives you time to go back and reread what you wrote. It gives you time to go back to some of those ones that you were like, oh, I don't really know about this one, and you want to get some extra points in there, or you want to guess on something. It gives you that extra time to go back. But specifically in questions 1 and 2, there's going to be multiple bolded terms. There's going to be multiple questions in there. If you give yourself a little checkbox, make sure that you checked off that you've done each component of that part. You won't skip any parts. Some people write the time on the page, like what time they need to move on. It helps them to know, okay, I've given my 20 minutes on this question. I need to move on and go to the next question. Order in knowledge or your ability. Don't order in the, okay, let me write question 1, then question 2. Order in knowledge, okay? And then when I say watch your question number, you're getting a book that just has lines in it, okay? It's going to tell you, start response question 1 here. Start response question 2 here, okay? So just watch that you're on the right page. Now, that's in full big letters, all that fun stuff. We can get your whole book. We will see your whole book. But it's just easier if you go ahead and have your question number. If you write it on the wrong page, don't freak out. Just like on the correct page, like see question 1 for question 2 response, and we'll be fine. We'll find it. Okay. So question 1 is going to be, a 8 to 10 point question. So part A is going to have 1 to 2 points on describing, explaining biological concepts. This is looking at your biology. Do you know the biology? Part B is 3 to 4 points, is identifying experimental design, control, null hypothesis, alternative hypothesis, giving you information, independent variable, dependent variable, stuff about the actual experiment. Part 3 is analyzing data. So they're giving you data, they're giving you a graph, and you're going to use that information and you're going to analyze it. Okay. And then part D is going to be making and justifying predictions. Okay. And so as I work through each of these, you'll see like a little box that will kind of tell you what supposedly it's about. You can kind of see the connections that they're making. So our first question is about this G-coupled receptor in the plasma membrane of the cell. We have intracellular signaling. So there's a GTP that is replacing the GDP that's bound to the certain subunit. So cyclic AMP is then going to get activated. It will activate protein kinase. In the cells, there's a certain process that's happening, which is then going to cause chloride to get released. And when chloride gets released, we all know that means that water gets released. Okay. They then tell you that individuals that are infected with this thing are going to have an overactive system. They tell you that we did this experiment. They tested 4 different samples. Some of them have GTP. Some of them have this toxin from cholera. And we're seeing how much cyclic AMP is produced, and you're looking at that data. So this is what they've given you for a long FRQ. You've got this big long prompt. You've got this diagram. You've got some data. So you're trying to then take all that in and answer some questions about it. Okay. So the first question we said is just straight biology. Okay. Do you know your biology? Okay. So describe one characteristic of a membrane that requires a channel to be present for chloride ions to pass across the membrane. Okay, so think to yourself, okay, chloride ions. Ions have a charge. They're positive, negative, it doesn't really matter, but there's a charge. Well, if something is charged, can it get through the membrane? No, it can't get through the membrane. So why can't it get through the membrane? Well, because the membrane itself is nonpolar. So talk about the membrane. Talk about how the interior of the membrane is nonpolar. There's these fatty acid tails, and these fatty acid tails inhibit the ability for this charged chloride ion to pass through the membrane. And so this is what they accept on the exam. Were you talking about the interior of the membrane being nonpolar? Did you talk about the interior of the membrane being not charged? Or did you talk about it being hydrophobic? You had to use one of these three statements about the membrane. So when they say describe the membrane, you couldn't just say plasma membrane is nonpolar. By saying plasma membrane is nonpolar, you didn't give enough information. So talk about the membrane, talk about the different components of the membrane that make it nonpolar. Talk about why it's nonpolar. Any of that is the description that you're needing. That's a little bit more than the identification. So it then says, okay, we'll explain why the movement of chloride ions out leads to water loss. Okay, well, chloride ions are some type of solute, right? So if there's a solute moving into an area, it's going to make that area more hypertonic. So there's more solute, less free water. Well, if the outside of my cell is more hypertonic, then logically the water is going to leave my cell and it's then going to move to that hypertonic area, okay? So you need to talk about how the space outside the cell was hypertonic compared to the inside when that caused the water to move out. Don't just tell me, well, the water moved out. Well, yeah, the question told me the water moved out. So explain why moving chloride ions causes waters to be moved out, okay? So you could have just talked about that there was a lower water potential compared to the inside of the cell. All of those were appropriate things you could have said. Most of the students on this one talked about hypertonic and hyperosmotic, not really as many said that one, but most of them said hypertonic. And you were able to say the opposite. As the ions move out, the inside of the cell becomes hypotonic, so the water moves out of the cell. That's perfectly fine to say that kind of stuff. So the second part, okay, now notice here that there are two different bulleted statements and then there's two different points here, okay? Oftentimes you can follow the bulleted statements to know how many points are going to be in the question. So when I was talking about the check boxes, I was meaning like, okay, check off the describe, check off the explain. That also helps you because when you, let's say you didn't answer explain, like you're like, oh, I don't know what to do on this one. And you come back to the question, you know you haven't answered that explain point because you haven't checked it off, okay? So B, identify an independent variable in the experiment. Okay, so when we're looking at this chart, right, what is the dependent variable? The independent variable is a thing that the scientist, the researcher is changing. So what did the researcher change? Oh, the researcher changed the presence of GTP. The researcher changed the presence of the cholera. Okay, so for this point, you would have said the presence or absence of GTP and presence or absence of cholera, okay? Well, actually, you could have said either, I'm sorry. So you had to talk about that it was either there or it wasn't there, okay? A lot of students talked about presence, but they didn't talk about absence, okay? So that was a big point that a lot of students missed on this question was that they didn't talk about presence or absence, okay? So do make sure that if you see a plus and a minus and they're asking you about independent variables that you say presence or absence of that specific material, okay? And you do need to be specific about the experiment. Don't just say, oh, it has the treatment group applied to it. Well, talk about the specific experiment. What in the experiment was the independent variable? So the second thing we had to do, identify a negative control. Well, negative control is talking about the group that does not have the treatment, okay? So if you look at this chart, what was not given the treatment? Well, the treatment we were looking at was GTP and cholera. So which sample doesn't have GTP or cholera? Well, that would be sample 1, okay? So you could have said sample 1, or you could have said the sample that lacks both cholera and GTP, okay? Now, they were super sweet on this one, okay? They gave you even more options. If you had said anything that lacks cholera, because that was one of our independent variables, right? So you could have said samples 1 and 2 are considered your negative controls because they lack cholera, okay? You could have said the sample that lacks cholera but contains GTP, which would have just been sample 2. Or you could have said the one that lacks GTP because that was the other independent we talked about, right? So that could have been samples 1 or 3. So literally it was wide open. You could have said almost anything and still gotten those points, okay? Yeah, based on somebody's in the chat saying, but doesn't the presence imply that something was not present, it would be absent? That was one of the big arguments that we were having at the read. So it depended on the way that you said it. So just to be safe, I'm telling you to make sure you write presence or absence. Most of the students who wrote presence didn't give the extra supporting part to be able to get that extra that point. Okay. So so as we said before, we've got the identified point, the other identified point, and now we've got to do the second justified point. Justify why the scientists included sample 3 as a control sample. Okay, so a lot of students told me sample 3 on this identification point right here. They're like, oh, it told me in the question that that's the control. Well, I told you that was a control. Like, so be very careful not to fall into that like bump right there. And someone in the chat says, can I repeat what negative control is? A negative control is going to be one that does not have the treatment applied to it. Okay. So it's making sure that the experiment is viable. It's making sure that the experiment is taking place, because I need to see, okay, if I didn't apply this treatment, what would have happened? Okay. So it's something to compare against. So this is justify why they used sample 3 as a control. Okay. So if I look here at sample 3, what do I see? Well, I see that it doesn't have GTP, but it does have cholera. So it's going to allow the scientists to be able to see, okay, well, if cholera is present, what's going to happen to the cyclic AMP production? Okay, so it allowed you to compare to sample 4, okay, because sample 4 has the GTP in it, and sample 3 does not, but they both have the cholera. So it gave you a point of comparison for the cyclic AMP to compare with having both cholera and both cholera and the GTP. Okay, so overall, you had to make sure that you mentioned that it was comparing to samples 3 and 4 together. Okay, so that was our third point for part B. So C says, describe the effects of cholera on the synthesis of ATP. Okay, so when we look at this, okay, well, we need to think about, okay, what do I need to look at to see cholera and cyclic AMP production? Okay, so you're saying, okay, well, cholera is here, cholera is there. Okay, cholera has no effect, because here we're seeing that is 0.5 to 0.5. So yeah, cholera has no effect. Well, you just fell into a trap, okay, because you weren't completely going through the entire question. Because if you look here at 2 and 4, you see that cholera in the presence of GTP has a positive effect, okay? It's going to amplify the amount of cyclic AMP released when GTP is present, okay? So if cholera is there and GTP is present, then we see an increase, okay? So cholera toxin increases the production of cyclic AMP in the presence of GTP, okay? So you had to talk about 2 and 4 together, okay? You could have also said the other way, right? Because we looked at before that if you looked at 1 and 3, it had no effect, but you had to stay in the absence of GTP. This is where a lot of students didn't get their point, because they didn't mention that presence of the GTP or in the absence of GTP, okay? And I scored this one on the AP exam. That's why I have so much extra information to give you about this one. I'll try to give you all the extra information about the other ones, but I didn't specifically score the other ones on the exam. So now it says, calculate the percent change in the rate of cyclic AMP due to the presence of cholera in sample 4 compared to 2. So when we're looking at this, they don't give you percent change on the formula sheet, okay? And that was a big hot topic for a lot of students. Like, I didn't know how to do percent change. Well, you should have been doing percent change in your experiments throughout the year, okay? Specifically, you did it in the osmosis lab, okay? Now, whether you remember that formula, I don't know, okay? But they do not give that formula to you. So let's talk real fast about what that formula says, okay? So percent change, okay? And I forgot to do times 100% over here, but the percent change is looking at final minus initial divided by your initial, okay? So we're looking at how did it change compared to how it was to begin with, okay? And as I said before, multiply that by 100 to get into percent, because I forgot to add that little part right there. I'm so sorry, y'all. So as I'm comparing 2 to 4, I'm gonna say, okay, 127 minus 10, and it was initially at 10, so I'm gonna divide by 10, and that's gonna give me 1,170%, okay? So teachers all across the board always teach you to make sure your number sounds logical, okay? If you saw that big percent, you're going to have a mini freak out because of the fact that that number is such a big percent. Okay. Trust your gut. Okay. If you know you did your math right, trust your gut. Okay. Because I know that is a crazy big number. Okay. So as I said before, we took our final of 127 minus our initial of 10, and we divided by our initial of 10, and that gave us 11.7, and we multiplied by 100 to get into a percent. Okay. So that was C. So on to D. Okay. So D is going to tell us to make and justify prediction. Okay. They're going to tell you something went wrong in the system. So you need to figure out, okay, after, like, because that thing went wrong, what do I need to do about that? Okay. So in terms of this, we're going to say, okay, well, a drug designed to bind to the cholera toxin. So it binds the cholera, it stops it from doing its thing. Okay. They mix the drug with cholera toxin, and then they add this mixture in the GTP to the different cells, predict the rate of cyclic AMP production in it. Okay. So they've given you everything you know. Okay. Looking at this chart, do you see something that has GTP that does not have cholera? Because of the fact that cholera is being bound, right? I don't have any cholera because the drug binds to it, okay? It would model sample 2. So sample 2 said 10, so my prediction is going to be 10. The rate will be 10, giving me, of course, my units, okay? They were nice on this one. If you didn't give the units, you didn't lose points, okay? But let's go ahead and leave no naked numbers on the exam. Let's make sure we always have a unit behind all of our numbers, okay? So the next part is that we need to say in experiment, the scientists engineer a mutant adenylyl cyclase that cannot be activated by the certain things. They claim that the cholera will not cause excessive water loss. So justify this claim. So they're telling you water will not be lost. Water will not come out of this system. Why will water not come out of the system? Let's look through that, right? So here I've got this pathway, right? They gave you the pathway. This is the only time we've had to use this pathway the entire time. They gave you this real ugly, graphic. You're like, oh my God, freaking out. But this is the only time we've had to use it, okay? So they say, okay, well, I don't have the adenylyl cyclase. Well, if I don't have adenylyl cyclase, what am I not activating? Well, I'm not going to activate protein kinase. I'm sorry, I'm not going to release the cyclic AMP. Well, if I'm not releasing the cyclic AMP, I'm not going to activate the protein kinase. Well, if I'm not activating the protein kinase, I'm not secreting the chloride. And if I'm not secreting the chloride, I'm not losing the water. Okay, and that is what you had to talk about. You had to talk about that the cyclic AMP is not going to get produced. And because cyclic AMP is not produced, protein kinase was not activated. And because protein kinase was not activated, chloride ions was not secreted. And then you could have just talked about the water. Okay, so now all of those statements had to be in the answer. Okay, you could just say, oh, there's no adenylyl cyclase, so protein kinase wasn't activated. You had to talk about the entire pathway because they gave you the entire pathway. Okay, I know it sucks, but if they give you a pathway, give me every single step on that FRQ. Make sure you explain it fully what is happening. Okay. So that's question 1. And clearly I'm not finishing this in an hour since it is only one question and I'm already 20 minutes into this thing. Okay. But I am going as fast as I can. And of course, you're probably, when you watch the playback, you're going to watch this on 2 times the speed anyways, right? So interpret and evaluate the experimental results with graphing. So part 2, we're going to have to graph and then we're going to have to analyze this graph. Okay. So number 1, describe, explain biological concepts. That's the biology. Do you know your biology? Part B, construct a graph. This is where you're graphing. And there's usually 3 or 4 points here, just being able to do a graph, okay? Part C is going to be analyzing that data, and part D, making and justifying predictions, very similar to A, okay? So during meiosis, we have this double strand break that occur in our chromatids. The break are either repaired by the exchange of genetic information between homologous non-sister chromatids, which is called crossing over. You all know this very well. Sometimes they're simply just repaired and they don't have any crossover. The breeding plants are going to develop new different varieties of corn. So we want to see if there are correlations between the number of double strand breaks and the number of crossing over. Okay. They then use staining. They give you all this information. You see that I've got, 6 different corn strains. They tell you how many double strand breaks they were, and they tell you the number of crossovers. Okay. So first question, again, do you know biology? The double strand breaks, along with the DNA backbone, describe the process by which the breaks occur. Okay. So y'all, let's not overthink this. Okay. Double strand breaks. Okay. How does a bond break in a macromolecule? Y'all, it's simple. Hydrolysis. Okay. So you just had to talk about that there was hydrolysis occurring between the sugars and the phosphates, or between the nucleotides. So talking about between different nucleotides, between the sugar of one and the phosphate of the other, there was a breaking of a bond. There was a hydrolysis that takes place. Great job to that person in the chat that says hydrolysis, okay? You also could have just said that the covalent bonds between the sugars and phosphates broken. You didn't even have to tell me it was hydrolysis. Just tell me that this covalent bond broke, okay? That's like, it was very simple, okay? And I'm gonna tell you right now, when I answered this, I was off in La La Land talking about all kinds of extra stuff. And Alice, I'm sorry it's 11 o'clock where you are. You are welcome to watch this later. Go to sleep, love. So B, using the template provided, construct appropriately labeled graph. Okay, so I'm going to go backwards real fast, and we're going to look here at this data. Okay, what kind of graph would I make with this? Okay, I have 6 different corns. Okay, they have different numbers of double strand breaks, and then those double strand breaks led to a certain number of crossovers. Okay, so what kind of graph would I make? Well, I'm going to make a scatter plot. Okay, so make sure that you know the kind of graph you're having. Okay, this one also threw students through a loop, especially my students, because they gave you the axes already labeled. Okay, so the axes were already labeled for you. Okay, and you would have just plotted your points. Okay, you plot all the points. And then, of course, you're going to give us the error bars on there. Okay. So notice that this person put a little break right here. You're allowed to break the axis before the data. Okay. So after the data, you can't really break your axis. And no, it's not a bar graph. It's going to be a scatter plot. Okay. Although this does look like a modified bar graph, this is considered a scatter plot. Okay. Notice also that they count each block is 1 in their aspect. They don't have to label every single one. They can skip. So here you see they skipped by 2. And they label their axes again down here. They're saying every 4 is 100. That is perfectly fine as long as you have a axis that makes sense for you to write it and like count easily. Then the readers can also count easily. You want to count by something that's easy. Count by 2, count by 5, all that fun stuff. So some people in the chat are asking, why is this a scatter plot and not a line graph? Okay, well, because these are 6 individuals. Okay, I'm not graphing the individuals on the axis, right? I'm graphing the double strand break, and I'm graphing the number of crossovers. Okay, those 2 things have absolutely nothing to do with the individuals being connected. They're just individuals. So you can't connect individual organisms, okay? Because you're not looking at a spectrum. Line graphs are spectrums, okay? You're not seeing a spectrum on this one, okay? So since there are 7 individual things, we have to plot them as individual things. Somebody's saying, well, how do I know the type of graphs to do? If you see sequential data, you're seeing linear data, that's a line graph. If you're seeing categorical data, like, I don't know, there's this part of a fish tank is 10 to 20, 20 to 30, 30 to 40, yada, yada. Like that's a range, that's like, you know, or you're doing, stuff that is super far apart. Like, so there was a bumblebee problem from a couple years ago where it was like 10 minutes and 24 hours. That is not a linear scale. You had to do that as a bar graph. So if it's categorical, you'll do bar, okay? So hopefully that was helpful for y'all. So the things that they were looking at here, they wanted you to have the appropriate axis. Did you have accurate error bars? So did you correctly put your error bars? Okay. Now, in terms of your error bars, this little plus or minus is how you know your error bar, okay? So you'll go 19.5, 0.5 up, and then you'll go from 19.5, you go 0.5 down. So you would have graphed this from 20 to 19 with a little dot in the middle at 19.5, okay? So that's how you would graph those. And then you had to appropriately graph it, X, Y with separate points and not giving me any type of line. I'm so sorry for making you panic. I'm not trying to make you panic. I'm trying to give you as much information as I can give you to help you be as successful as I can help you be. Okay. Sorry, y'all. Okay. So based on the data, determine whether strains 1, 2, and 3 differ in their average number of crossovers. Okay. You'll connect the dots if it's a line graph. So if I look at 1 and 2, okay, notice the error bar does not overlap. Okay, so I know it's small, it's a very little itty bitty bit. Okay, but that error bar does not overlap. And so since the error bar does not overlap, 1 and 2 do have a difference. Okay, they are different in the number of crossovers. Okay, 1 and 3 also do not have any type of overlap of their error bars, okay? And so those ones also differ, okay? But 2 and 3, which is why I'm making it red, they do overlap, okay? So you would say that 1 and 2 do differ, 1 and 3 differ, but 2 and 3 do not differ, okay? And so that's what they were wanting here. They said there is no statistical difference between 2 and 3. Strain 1 is higher or is different compared to 2 and 3, okay? So you had to give that full information. I didn't score this on the exam, but that is the full information you had to have. There's a couple questions in the chat about the graph. So here you see there is 2 little lines there that say that they broke the graph, okay? Every block means 1, okay? And so by breaking that axis, it's telling me, okay, well, between here, it's not going to be 1, but that's okay because we're breaking the axis, okay? Do we need to label the numbers for the error bars? You don't need to label the error bar numbers, but you did need to label your organisms. So you see how they labeled 1, 2, 3, 4, 5, 6 for the different organisms. You did have to label your organisms because of the fact that this was a scatter plot, okay? Do we need to label the number? Okay. So I think we're good there. So based on the data, describe the relationship. So we have this chart, we have this graph. We need to say what's the relationship between the average number of double strand breaks and then the average number of crossovers. Okay, so as you see in your graph, it kind of makes this like upward trajectory almost, right? So they're going to be directly related. Okay, there's a direct correlation, a positive interaction between them, between the number of breaks and the number of crossing over. You can clearly see that the more breaks there are, the more crossover is taking place. There's a direct relationship. Great job, Sarah. Okay, so D, looking at crossing over. Ooh, okay, so this one threw a lot of students through a loop. Okay, and I made a little diagram to try to help us out, to try to visualize what's happening. I am not the best when it comes to making these little diagrams. There's definitely better people out there to make diagrams, but I did my best for y'all, okay? Great job, Luigi. So it says that crossing over creates physical connections that are required for proper separation of homologous chromosomes. Now, this is one of the things. Something went wrong. Now give me a prediction, okay? So a diploid cell with 4 pairs of homologous chromosomes undergoes meiosis to produce 4 haploid cells. Crossing over occurs only between 3 of those pairs, okay? Now, it clearly said right here that in order for us to have proper separation of homologous chromosomes, they have to cross over, okay? And this is where students didn't notice that little piece of information and they went based on what they know, which is great. I know, I'm so happy that y'all know so much biology, but you didn't use the question prompt. This is going to talk about something went wrong, okay? What went wrong? Went wrong because the fact that 3 of them are the only ones that had crossover take place. So here are our 4 homologous chromosomes. I made them different colors just so we could easily see them and follow them through this little graphic I made, okay? 3 of them had crossover take place. Awesome job, yay. So yellow did not have crossover. I'm not going to switch the sides and make them pretty, but I hope that you can remember that those ones crossed over, okay? So when we go through meiosis 1, our homologous chromosomes, they separate. They go through anaphase 1, they segregate. We see them moving into 2 different cells. But the yellow one did not have crossover, so it stays together as a unit, okay? And so at the end of meiosis 1, I have this cell that has these 3 homologous pairs, or these 3 sister chromatids of whatever chromosome, okay? And then over here, I have these 3 sister chromatids, and then this 1 homologous pair that's still together because it couldn't separate, okay? So when I go through the second round of meiosis, okay, of course, let's look at the right side, because it's a little bit easier to follow that. My sister chromatids segregate, they come apart, so we're easy there. So there's 2 cells that will have 3 in them, okay? Now, the other 2 cells, now our sister chromatids come apart. So I separated them just so you can see like these sister chromatids came apart and these sister chromatids came apart. So I put the green one in the middle just for visualize, okay? So these 2 cells are going to have 3 and these 2 cells will have 5. So the correct answer here would have been 2 cells with 3 chromosomes, 2 cells with 5, or you could have said 2 cells that had n-1 or 2 cells with n+1, okay? Now, I know that's a little hard to wrap your mind around, okay? So as you see here, I drew a picture. And if I was doing this on the exam, I would have drawn a picture. You are fully capable and able to draw pictures on the exam, okay? What you need to do when you do draw a picture like that is just label the diagram. Just say, as seen in figure 1, I have 3 in 2 cells and 5 in 2 cells, or however you need to say it. Make sure you label the figure and that you refer to the figure when you do your explanation, okay? And so then it said, of course, just predicts, justify your prediction. So here's our justification. 3 of them separate normally, 1 pair does not separate, and then in meiosis 2, the sister chromatids separate. Okay, so I hope that my little visual kind of helps you out. When they say n-1, they're saying like if our chromosome number is 4, it's 4 minus 1, which is 3. If our chromosome number is 4, so n+1, that would be having 5. Okay, so that was really something. So I can submit pictures along with your written response. Yes, you're perfectly able to, draw a picture and then like refer to the picture. Okay. Yes, you're completely able to draw pictures. Okay. So the second part of this question, explain how the plant breeders use the information in table 1 to help develop new varieties of corn. Okay. So table 1 told us that chromosome double breaks and crossing over means that there's a correlation, right? The more double breaks there is, the more crossing over. Well, they're going to find some way to increase genetic diversity by causing there to become more crossover numbers. Okay, they will increase the double strand breaks, leading to more crossovers. They're going to breed ones that have higher crossover numbers. They're going to make it so that they have that more genetic diversity due to those breaks. Okay, so that was, again, there's a point. Where do we draw the picture? On the lines. So like in your book, you're going to have all these lines. So just draw the picture on those lines. Don't try to smush it in between the lines. Just like draw yourself like a little box and then draw your picture inside the box and just ignore the lines. I've actually never seen the book. When we see the responses, there's no lines on it, although I assume there's lines. I'm assuming they're probably like very faint lines in your book. And so you probably could ignore the lines easily. Okay. So that is our 2 long ones. So moving into our short FRQs. Okay. So we have the first one's going to be a scientific explanation. Okay, this will be worth 4 points. Notice that each part is worth 1 point. So you can literally just think about, okay, let me make sure I answered all A through D. Okay. So A is, again, do you know biology? Describing biological concepts. B is identifying experimental procedures. C is predicting things, and then D is justifying things. And a lot of times students marry C and D. So if you accidentally are like writing C and you just end up justifying it, you don't need to rewrite your justification. Just put a little D near your justification, and that will help us to ensure that we find your justification. Okay. So we have these fruit flies. They emit light. Okay. When there's the enzyme luciferase catalyzes the reaction in which the substrate, they react to form this certain chemical and these other products. In order to determine the optimal temperature for this enzyme, they add ATP to the solution containing the thing. And then they're going to measure the amount of light emitted during the first 3 seconds of the reaction when it's carried out at different temperatures. So they're watching what's happening due to the temperature. So part A says to describe a characteristic of the luciferase enzyme that allows it to catalyze the reaction. So how does an enzyme speed up reaction? Well, it speeds up reactions because it's going to bind to a specific active site. Okay, the substrate binds to the active site, and by binding to the active site, it allows those substrates to react more efficiently. It puts them in the right orientation, and so all of these things are decreasing your activation energy. Okay, but you had to talk specifically about the enzyme and how the enzyme decreased the activation energy. So it was mostly just talking about that the things bound to that active site. What is the difference between long and short FRQs? Long FRQs are more points. They're 8 to 10 points. They usually have a longer prompt. Short FRQs are only 4 points each. And so there's just shorter prompts, less you're going to have to write for them. So usually I spend about 8 minutes on a short FRQ and about 20 minutes on a long FRQ, which we're kind of following that time period because it's 4:40 right now. So hopefully I can do these in 8 minutes and I will fit within my hour. So that'd be kind of cool. Actually, that would fit in an hour and a half. Whatever. I'm going to try my best to finish on y'all. Someone says lowers activation energy. Yes, enzymes do lower the activation energy, but they want to know a characteristic that allows it to catalyze the reaction. So what is it doing to decrease that activation energy? Sorry, y'all. So identify the dependent variable. Well, earlier we talked about independent variable. So now we want to talk about the dependent variable. The dependent variable is the variable that is changed due to the independent variable. So because the experimenters changed something, because the researcher did something to it, what is the thing that we're getting out of it? What is the thing that's being measured? What is the result? Okay. So when they ask us to explain and describe, you say prior knowledge. Yes, explaining and describing are bringing in prior knowledge and applying to the question. Okay. So identify a dependent variable. Okay. So when I look back at my prompt, I'm like, wait a minute. They measured the amount of light emitted. Oh, well, then it's the amount of light that's emitted. Yes, this video is going to be posted on Marco Learning's page, and then I'll copy it, of course, to my page like I always do. So the amount of light emitted is the dependent variable that we have here. Okay. That's a simple question. Okay. Now, a lot of teachers you have probably tell you that if it says identify, you could just write the one-word answer or the short little blurb. And so that is true on some questions. Okay. So I always tell my students, just write it all out. Okay. The variable is the amount of light emitted. Like, it is not that hard to write those 3 extra words and ensure that you get points. Today we're not doing multiple choice because today's designed as a FRQ only session. That's why it's called FRQ review. So that's that. Okay, so I'm going to answer that question in just a second. But so C says state the null hypothesis. Okay, so the null hypothesis states that the treatment had no effect. Okay, but you can't just say the treatment had no effect. You got to be very specific for our experiment. Okay, so we're saying that the temperature should cause a difference in the amount of light. Okay, so your null hypothesis is going to basically say there is no effect. Temperature has no effect on the amount of light emitted. Okay, so because of the fact that that's what we're testing, we're saying if I increase temperature, there should be a change in light, or I expect there to be a change in the light. Well, null hypothesis says that the temperature has no effect, so there would be no effect on a change in temperature on the amount of light that is emitted. Okay, so then part D says, a student claims that as temperature increases, there will be an increase in the amount of light given off by the reaction in the first 3 seconds. Support the student's claim. Okay, so as temperature increases, there's going to be an increase in the amount of light given off in the first 3 seconds. Well, what do I know about temperatures and reaction rates? Well, if I increase temperature just a little bit, like not a lot, I'm not saying like huge amounts, but a little bit, it's going to increase the kinetic energy of the molecules. And I know that hits a little bit of chemistry, but we assume you know a little bit of chemistry. Okay. So the kinetic energy of the molecules is going to speed up, which is going to increase the frequency of the collisions of the molecules, which will increase your reaction rate. Okay. Also, higher temperature results in a change in the active site, and that can enhance the substrate binding. Okay. So those were the 2 different things you could have put here because they're asking you to support. They're telling you the claim is right. Now tell me, why is the claim right? What about what's happening inside of this reaction that makes their statement correct? Okay. So somebody says, do I recommend doing my short FRQs first in terms of take of time? No. I recommend that within your, well, you can do them first, I don't care. But within the first the first 3 FRQs you do, I want there to be 1 long and 2 shorts. Okay. I want you to at least get one of your longs out of the way. And I want you to at least label your axes, because it's very easy to do the graph. Okay. At least get that point done. Isn't it chi-square? We haven't done anything with chi-square right now. Okay. Okay, sorry, y'all. So FRQ4 is conceptual analysis. Describe biological concepts. Again, do you know the biology? Okay. So a lot of y'all are going to be like, oh, Miss Jones, I don't really know the biology. I've struggled all year. Skip A. Just go straight to B. Use what you know. Use the prompts. Use all this information to answer the lower questions. And then if you have time, go back to the A. It's fine to skip around. Okay. Answer them in the order you know. Okay. So part B, explain biological concepts. Again, that's another biology part. C, predict the cause and effect of a change. And then, of course, your justification. So we have this brook trout population in Newfoundland, Canada. Once part of a larger population, they were fragmented at the end of the most recent glacial period. Researchers investigated 14 naturally separated stream populations of brook trout. They found the population are all genetically distinct and show differences in morphology. Okay, describe the prezygotic barrier that results in these genetically distinct populations. Okay, prezygotic barrier. A prezygotic. Let me break down this word because I don't know what prezygotic means. Okay. Pre means before. Zygote or zygotic, I think of zygote. Okay. Zygotes are the things that happen when sperm and egg come together. It forms a zygote. So it's the new thing coming together. Okay, cool. It's the new organism. So something that's going to stop a sperm and egg from coming together. Oh, that's easy. They're in 2 different places. So geographic isolation prevents the gene flow between these populations. So we're seeing that they're genetically distinct because of the fact that they're in different streams. And because they're in different streams, they're no longer mating with one another. So there is an absence of that gene flow. You do not have a 10-minute reading period, so your teacher is wrong. Your reading period is embedded into your 90 minutes. So you're welcome to take 10 minutes and read the questions, but you only have a total of 90 minutes for all of your FRQs. Right, so habitat isolation, I don't remember if that was accepted on this one because they're all living in the water. We specifically, I think, had to talk about them being in a different geography. I was not the reader on this question, so I don't really know if habitat was accepted, but it is a logical thing to say that, but this one specifically was looking, I think, geographic. Does it geological have to do with time period? Geological is a different time, so no. So here I say, okay, well, brook trout with longer fins are able to swim faster than the brook trout with shorter fins, okay? In one of the Newfoundland streams, the main prey evolved to move faster. For brook trout living in the stream, explain the difference in fitness between the longer finned individuals and the shorter finned individuals, okay? So if my prey moves fast, I need to move fast. So the difference in fitness, because fitness is just looking at reproductive isolation. I'm not reproductive isolation, reproductive success, right? So if you're more likely to survive, right? Because you're more likely to reproduce, and you're more likely to have offspring that are more likely to survive and reproduce, that means you're more fit, okay? So the longer finned individuals can swim fast and they can get food. So they're more likely to get more food. They're more likely to survive and they're more likely to reproduce. So that is why they are more fit than the shorter finned ones. Okay. So individuals with longer fins, more likely to capture prey and reproduce. Okay. You had to talk about both parts of these. You had to talk about that they could get more food to survive more. And you had to talk about the reproduction part. The fitness always involves survival as well as reproduction. So you have to make sure you bring all that in. Okay. So C, two morphological and behavioral distinct populations of brook trout remain isolated for many generations. Predict the impact of both populations, okay? But so if they are isolated for a long period of time, logically, they're going to become more and more genetically distinct to the point that maybe they won't be able to mate anymore. That is called allopatric speciation, okay? If two kind of populations are in different areas or in the allopatric is different areas, okay? They're geographical barriers and they no longer mate with each other. They're going to create new species that can no longer mate with each other, okay? So the two populations will become separate species. Two populations will continue to diverge. They'll either become different behaviorally, morphologically, or genetically, okay? So you did this these are the 2022 exam questions. Your teachers definitely could have probably put those past FRQs on tests. I do the same thing, so it's logical you would potentially see these questions. I just wanted to go over them so you had like a good explanation of all of them. Or at least I think that's a good explanation of them. Researchers claim that there's more genetic differences between any two brook trout populations than there are between any single current population and the ancestor, okay? So that's saying that there's more differences between these two populations than there is between the population and the original ancestor, okay? And they want you to justify that. Well, because of the fact that these two are in two different areas, okay, they have mutations that are occurring in one that's not occurring in the other, okay? So a single population could accumulate these mutations, okay, or they're going through that, of course, genetic drift, and that, of course, accumulates, which then leads it to a difference, okay? Another option you could have put here was just about the allele production. There's these random mutations that lead to new alleles, and because of those different alleles, there could be a different amount of selection that took place that leads to the divergence or the difference in these two different populations from each other, okay? So let's talk about some of the stuff in the chat. You wouldn't have to mention allopatric speciation. It's perfectly fine not to, because of the fact that the question you didn't have to talk about allopatric. You just had to say that they become separate species. But it would have been, good if you would talk about allopatric. Somebody says their bio teacher hasn't started reviewing. That's perfectly fine. Some teachers haven't finished content. We're all in different time periods. We all start at school at different times. you were the one taking the exam, so it's your responsibility to kind of start reviewing on your own. We've got 11 days for the exam. So, find some YouTube videos. There's a ton of mine that are on Marker Learning from 2 years ago that you're welcome to look at. I've got all the TikTok videos you're welcome to see. There's tons of resources out there. So you could just start looking over the content that you're weak on. All right. So FRQ 5, okay. If evolution is caused by mutations, and how do animals get advantageous traits? So mutations are due to a random change in the DNA. And so based on the environment, one trait could be more favorable than another trait. And maybe that trait wasn't there before until that mutation took place. And that's why we see the advantageousness taking place. Okay. So analyze the model or visual representation. So 5, you're going to have to do a picture. You're going to have to draw something. Okay. So part A, describe characteristics of a biological concept. B, explain relationships between different characteristics. C, we're going to do the drawing of representing relationships. D, explain how the concept goes to a bigger principle. So you're going to connect this somehow in a bigger span. Okay. So these models represent interacting species in different communities. Some of the same species and feeding relationships. These models assume that both communities have the same initial biomass. Sorry, y'all. The models can be used to understand the effects of human activities on the community. So here we see community A, has a ton of these different primary producers, a lot of primary consumers. There was a big web going on here, okay? And community B is a little less, right? You got 2 primary producers, and the primary consumer only eats that primary producer, and same thing here. And so there's less kind of going on here, okay? So A says, describe a characteristic of the community that makes a species invasive in that community, but non-invasive in a different community. So this has absolutely nothing to do with these diagrams that we just saw, but what about one community would make an invasive species able to thrive while another one not able to thrive, okay? Well, it all has to do with that they have an unlimited amount of resources, okay? So in this community, maybe there's no natural predators. Maybe there's no competition. Maybe there's, an ample amount of these resources. There's no diseases. So all these different things that would cause a invasive species to be more favorable in that community. It all has to do with the interactions of those other organisms, okay? So you had to connect it to the other organisms, okay? Yes, a week is enough time to practice. You can watch tons of videos. Look at College Board daily videos. Yeah, they've posted 2 different groupings of videos, so you've got all that. So B says, explain why removing primary producer 1 will have a greater impact on community B than community A. So if I take out the primary producer 1, so get rid of primary producer 1, it's gone, okay? What do you notice about the organisms that eat the primary producer 1 here? Well, you still have primary consumer 1, that one's probably going to have problems. But primary consumer 2 can still get food, right? So if I look here, primary consumer 2 still gets food. But over here, the primary consumer 2 has no food, right? Their food is gone. We removed that food source. So community B is going to have a disastrous effect because now primary consumer 2 has no food. So we're going to see a decrease in primary consumer 2, right? Which means that we're going to have a decrease in secondary consumer 2, right? And so you're seeing that there's a huge impact because of the fact that one organism was taken out, okay? So community B, there's going to be a decrease in all those different organisms versus in community A, primary consumer 2 has another form of food. You talk about that there is less diversity in our primary producers, so there's fewer paths for the energy to move through the community. Talk about how there's fewer species, fewer speeding interactions, there's less diversity, so there's going to be less resilience when there's a problem. Does this cause a increase in PC3? I don't think it would cause an increase in PC3. I guess if PC, these 2 aren't competing, so I really wouldn't expect there to be an increase in PC3, only because I guess you lose PC2. I don't know. That'd be interesting to think back on later. So good idea to include more than one explanation. No, answer only once. Don't try to keep adding things because then we just think you're pulling at straws. We only grade the first answer you give us. Don't give us more than one answer. In invasive species, each individuals of species SC2, okay, is introduced into the community using the template provided in your response for community B. Indicate the feeding relationship for the invasive species by correctly placing INV to represent the invasive species and an arrow to represent the feeding relationship. Okay, so here we see SC2. Okay, we know that it eats SC2, so logically we're going to find that the invasive species is going to be somewhere here, and there's going to be an arrow going from SC2 to that INV. Okay, and this is that appropriate answer. Okay, so you had to make sure that you put INV somewhere close to the TC2 and that you have that arrow going toward it. Okay. Okay, so D, explain how human activities that add toxins to the soil could change a community with many species and each trophic level, such as community A, into a community with fewer species and each level like B. Okay, so basically we're saying, okay, if I add toxins to the soil, why would this one become like that one? Okay, well, number one, biomagnification. Okay, if we have more toxins in the soil, okay, these toxins are going to go moving up the food chain, up the food web, okay, in these higher organisms, and they're going to have biomagnification, which is going to cause a decrease in them. Okay, you could also talk about that these toxins could be eliminating our primary producer, and by limiting that primary producer, we're reducing our species diversity at those higher trophic levels because they consume those primary producers, and so it could end up becoming more like community B. Okay. So I hope that that was helpful. I see there's some good conversation going on in the chat, debating those answers about the question B. So number 6. 6 has to do with analyzing data. So they're going to give you some type of data, and you've got to be able to describe that data and then evaluate a hypothesis or a prediction, and then explain how that relates to some type of larger biological concept. Okay. So this question was a really interesting one. Okay. In which we had an RNA vaccine designed to protect individuals against certain diseases. They developed these vaccines. They remove it from the individual. We then use the mRNA to code the pathogen, and then we make these certain proteins. It produces an immune response to those proteins and helps them in the future. And then they introduce these cells, the mRNA used in vaccines must be stable, so they're not degraded. They then talk about using these different caps on them, and how those caps can then kind of protect those different mRNAs, okay? And then they give you some data. They say, okay, well, this one has no cap, ha, ha, ha. This has a normal GTP cap. This one has a modified cap, and there's these 3 different, caps, okay? They then tell you the half-life, and they tell you the amount of protein that's translated from that mRNA, okay? So the first question we have is, based on the data, identify which cap structure is most likely to protect the end of the mRNA from degradation. Okay, so if a mRNA is degraded, that means that it's going to kind of get shorter. Okay, and so it's going to have a shorter half-life because it's going to degrade it faster. Okay, so if we're looking here, we see the one that has the longest half-life is 27. Okay, and so we can say, okay, well, the one that was protected must have been modified cap 2 because of the fact that it had the longest half-life. Okay, now it says you just had to identify, you don't have to give any explanation. So I would just say modified cap 2 protects the mRNA. Like, add those 2 words, ensure you get the point, all that good jazz. Okay, so that was your answer there. Okay, and then B, based on the data for the mRNA, describe the relationship between mRNA half-life and the total amount of protein that's produced. And somebody in the chat is asking a funny question. So they said, I wonder if they're talking about the COVID-19 vaccine here. And actually, because I was at the reading, I heard the whole story about this. This was the question that was supposed to be given prior to COVID drama happening. Or actually it was written before that. I don't remember if it was actually supposed to be on the exam or it was just written before that. But this question was written back in 2018. It may have been 2019, but it was back before COVID happened. So it was interesting that this question showed up because it was not actually written due to COVID. Okay, so based on the data, describe a relationship between mRNA half-life and the total amount of protein produced. Okay, so I'm looking for a relationship between this and this. Okay, so what do I see? As I increase half-life, what do I notice about the amount of protein? Okay, they are directly related. A longer half-life is associated with more protein, or there's a positive relationship, a direct correlation. All that stuff would help you to say they interact with each other, okay? And all you had to do was look at your data, okay? So as I said before, if you don't know A, just move on to B, okay? Use the data to help you. Use your prompts, use your diagrams, use whatever they're giving you in that question so you can try to guess, okay? And even if you don't know an answer, it's saying to describe a relationship. It's either going to be positive, negative, or no effect, okay? Try one of the three. It's got to be one of them, okay? And if you accidentally get it right, hey, that's a point, okay? So C says, after examining the data on the mRNA half-life, the amount of protein produced, researchers hypothesized that each mRNA molecule with modified cap1 was translated more frequently than each mRNA molecule with normal cap of GTP, okay? So I need to look specifically at the normal versus the modified, okay? Both of these two have approximately the same mRNA, but yet they have different amounts of protein, okay? So evaluate their hypothesis, okay? They're saying the cap1 is more frequently. Based on this data, I would agree. The data supports this, okay? It supports this because I see in modified cap1, there's 4, but in GTP, there's only 1, okay? So make sure that you are explaining, okay, what's happening here. The data supports, okay? If they're asking you to evaluate, you must tell them support or you must say refute. You need to say either one of those words. Either tell me you agree with it or you don't agree with it, okay? Give me something to tell me whether you agree with it, okay? Don't just tell me about the hypothesis. Don't just tell me about the data. Tell me, do you agree? Do you disagree? Okay, so it supports the hypothesis because the half-lives of the mRNA are the same, but the amount of protein produced with one is more than protein than the normal cap, okay? Or saying it's 4 times as much was perfectly fine to say that, okay? So D, introduction of mRNA into cells allows the cell to produce foreign proteins that might not normally produce. Explain why the production of foreign protein may be more likely than the introduction of mRNA than DNA. Okay, think for a second. Where is RNA? mRNA is in the cytosol. Where is DNA? The nucleus. So if I inject DNA into a cell, can I do anything with that DNA? Nope. The DNA has to get into the nucleus for me to be able to do anything with it. Okay. So by injecting an mRNA, I've got the ribosomes there to go ahead and start translating it to be able to make those appropriate materials. Okay, so protein production from the mRNA requires factors to initiate transcription, which is not going to be found in the cytosol. Protein production from the mRNA does not depend on the processing of pre-mRNA, so you don't have to talk about cutting out the introns that would have been in there due to the DNA. And the cell might be unable to transcribe the DNA while mRNA is directly translated. So you talk about how the DNA is not going to take place. Okay, so that brings us to the end of all of the FRQs from 2022, okay? So do you need a little bit more help with your FRQs? Do you need a little bit more practice with them, okay? So on my website, apvalpenguins.weebly.com, okay, there is a 351-page review guide. I'm not telling you to do the whole review guide. We only have a week, okay? Well, a week and a half, but whatever. So if you go into that review guide, it's broken down by units. At the end of every single unit, there's FRQs specifically for that unit. I've already told you where those are, okay? So all you have to do is say, okay, well, I'm really struggling on unit 5. I need to do some unit 5 FRQs. Go to unit 5 and practice your FRQs. The FRQ is there as well as the scoring guidelines. These are all College Board questions. I am not like making up my own FRQs. I just organized them for you, okay? Do you want to play some review games? I have some review games in which you have to figure out what kind of graph is it? Because I know we all struggle with, is it a bar graph? Is it a line graph? Is it a scatter plot? What kind of graph is it? Okay, I've got graph ones. I've got analysis ones. They're giving you a data set. You need to analyze it. What are you seeing based on the data that you have in there? Okay, and then task verbs. Maybe you don't know what the task verbs mean, so I need to, revamp myself on those different task verbs. So I've got the games for those. Okay, there's FRQ Fridays, and I know there's a lot of numbers in here. So on my website, there's a bunch of different. Actually, I can just show you. 1 second. So on my website, okay, you're going to see it says previous exam review. Okay, so if you go here, you're going to see that under 2020 and 2022, there's an FRQ Friday. FRQ Fridays for the 2022 exam year. Those ones, there's 12 different ones there. And I know it's kind of slow. Internet's a little weird because I'm currently online. But so there's 12 different ones. They're not the best quality because I went on Instagram Live for those, but they do exist, okay? If you go to 2021, 2022, there's again FRQ Fridays. Those ones are only me going over the 2021 exam, okay? So there's 4 of those videos. This one's the first long FRQ. This is the second long FRQ. These are 2 short ones and then 2 short ones. Again, not the best quality because of the fact that it is on Instagram Live. But again, it still is a resource if you're wanting to hear someone going through FRQs. You need to hear the logic that goes behind it. Yes, the review games are also on that same website. So sorry, y'all. If you go under, here's where like everything is for this year. So right here, you'll see all the game codes. There's like 120 different games in there. So like, you know, here's like specifically topic ones. But if you scroll down, there's ones like based on the CED. I like combined a whole bunch of the games. And then here is the Insta review. So for the past almost 3 months, I've done reviews on Instagram Live, on Instagram stories. So these are all the questions from those. So like you can look at a certain topic and there's all the questions that I did for those topics. And so you can kind of use those to break down. Okay, so I'm telling you, there's a ton of resources on here for you, all ready to go. Okay. So I was gonna say more study tools. That's all the stuff that I have. Okay. I also have TikTok videos for every single unit, going up to 7.5. Sorry, we hit February and I no longer had like time to do all the extra ones, but I did make topics 1.1 through 7.5 on TikTok format. I've got review PowerPoints on there. I've got videos. I've got 7 videos that I did with Marco Learning 2 years ago, where I go through every single unit. All that is available to you. Just if you honestly just go to Marco Learning's YouTube page, you're going to get to all those videos there, as well as their links on my page. Like everything I can give you, I'm giving y'all. And I'm going to go live. We haven't decided when I'm doing it. I think I'm going to do it on Sunday at 4 o'clock. I'm going to do a cram session. I don't believe in cramming the night before, so I'm going to do the cram session on Sunday. I'm thinking 4 o'clock, same time I'm doing this one. I'm just on a Sunday, not a Saturday. And so make sure you follow all the fun accounts. And that's all that I have for you today. Sophia, you're welcome to log us off. And remember, y'all, AP by page just steps by y'all. Sophia. Maybe I'm ending this thing. I don't think I can do this. I don't think I can end it.